in my daily life. So I was originally trained as a theoretical physicist in the Netherlands. When I finished my master's, which I did with a string theorist, I decided I didn't want to do such things anymore. I moved to the US, to Santa Fe, New Mexico, and I started working on theoretical models of evolutionary dynamics, sort of complex system theory. And like other people, I wasn't trained as a statistician at all. When they taught me physics, they assumed, you've got some probability, you've got some stochastic processes, obviously, but the sort of statistics and putting error bars and things and so on, this stuff you just pick up sort of by osmosis. You don't really need an extra course for that. So that's what sort of my state. I did always have some sort of problems with statistical mechanics. I remember even from high school that when they first taught me statistical mechanics, and after they explained me that all these beautiful classical... All right, that works. All right. So, yes, I don't know if others of you have had this too, but you get these classical Newtonian kinematic laws. It's made clear to you that these are time reversible laws, and then they tell you that actually, if you take a box of many of such balls undergoing such bouncing around, then there is actually a quantity that goes in one direction in time, and nothing the other direction, then the entropy. And I mean, I remember that even in high school, I thought this cannot be, right? Something is unreversible, and it cannot have this kind of asymmetry. So it always bothered me, but of course you forget about it, you just deal with it. And then when I was in Santa Fe, I think I was sort of there for a year, I had a new desk. And somebody left a large pack of printouts in this desk that they had, that downloaded some file, printed it out, put it in a folder, and left it. To this day, I don't know who left it. But it said, Probability Theory, the Logic of Science, E.T. James. And when I first read this, I thought, sounds a bit crackpotish, actually, this title. Probably, in Santa Fe, there were quite a lot of crackpots, so I thought this is probably just another one. So I didn't look at it. So one day, when I was sort of had nothing else to do, or I didn't feel like doing anything else, I started sort of browsing through. And actually, I quite quickly got hooked. I thought this is actually really interesting. And I think now that probably this, so I want to make a big plug for this book. So Ed James put this, as he was writing it, put it on the web. And but unfortunately he died before he could finish it, and then after a while, one of his ex-students, Larry Bradhorst, edited it and made a finished version, so you can now buy it in this book. So this is called, try to use this thing functionally, right? Probability Theory. Logic of by E.T. James. And luckily for me, Linda has made a nice introduction into the basic ideas. I think for me, what was very compelling is that by looking at probability as an extension of essentially propositional and Boolean logic. So, propositional logic is basically a way of how you do calculus with statements that are either true or false. And it goes sort of almost back all the way to Aristotle. And if you now say, well, I'd like to extend this, beyond looking at false and true, I want to assign numbers between 0 and 1, where false is 0, true is 1, but I want to also be able to give arbitrary sort of plausibility numbers between 0 and 1. And now I want a calculus that reduces to logic and that has some basic consistency requirements. Then using these theorems of Cox, you can make a quite compelling argument that there is really only one solution, and this is what we know as the standard laws of probability, Bayesian probability. But if you view it like this, then it becomes a very much more general thing, and it's very nice that statistics, statistical inference, statistical mechanics, maximum entropy methods, all these things are just different facets, different applications of this very general theory. I find this very, very compelling, and so I started using this in my work. One of the things was I was working on this evolutionary problem, and I had come up with a way of making nice, sort of relatively nice theoretical predictions by representing the state of the evolving population by some macroscopic variables that I thought were the most relevant ones, and then I intuitively just used maximum entropy, and it worked very well. And I remember my advisor telling me, okay, but you have to argue why you can use maximum entropy, because this is not some thermodynamic system in equilibrium. It's a very, very different kind of system. And so I tried to make some ergodic argument how this thing would randomize, so it would never work. In fact, I could easily show that the maximum entropy distribution is wrong. It's not the actual distribution. All right? But for the purpose of predicting the behavior of the macroscopic observables, it worked perfectly well. Because if you use the maximum entropy distribution, what you do is you just take a distribution that assumes nothing beyond the state of the macroscopic variables. And if that is enough to accurately predict the dynamics of these macroscopic variables, it means you do not need additional information. And so the only way you could sort of go really wrong is if the real distribution was biased in some very specific way that would affect the macroscopic variables. All right. So this is how I got into this thing. And during my PhD, I started working on problems in biology. The first genome sequences came out. I started getting very interested in this. The first gene expression microarray, the first Affymetrix prototype chips came out, and I was very excited by these possibilities to really measure states of biological cells in this very comprehensive way, to know their whole genome. And I started working on these problems, and this is sort of the field that I've been working on ever since. Computational biology, computational systems biology, genomics, this kind of stuff. So what I wanted to do, before my computer died on me, is to give you a number of examples of the kind of problems we're looking at and how we use Bayesian ideas, information theory ideas, to look at these problems, to analyze this kind of high-throughput data. Now, so what I will just try to do now, and you'll just have to bear with me, I don't know how well this is going to work, I will just take one of these problems where I think we had a nice Bayesian solution, and then just try to explain this problem, how we solve this problem. And to do this, I have to, I guess, introduce a couple of basic facts about biology. And Rico Fermi famously said that you should always, when you give a talk, assume your audience is infinitely ignorant and infinitely intelligent. And so this is the assumption I will do. I know many of you are not biologists, and so I will just start by telling you what I think are some relevant facts to understand the problem we've worked on. I know some of you are working on these areas, so bear with me. Maybe you find some of the summaries of what are the key points somewhat interesting. All right, so I think the first point is evolution. The entire theoretical framework for understanding anything in biology is evolution. Darwin's theory of natural selection. And I think the key point for us here is that it had, even before Darwin's time, long be recognized that you can very naturally divide organisms into a sort of a tree-like hierarchy, right? So you have a particular kind of lions that may be, you can think of that as one example of the sort of lion-like creatures, which is one example of cat-like creatures, which is one example of mammals, which is one example of, you know, bilaterally symmetric animals, and so on. You know these things, right? You have this Linnaean hierarchy that was drawn before people knew anything about evolution. And I think the key insight of Darwin is that if you draw a tree like that, that actually going from the root to the leaves represents time. Right? So there's some key insight that as you go from the leaves of the species living today up this hierarchy, you're actually going back in time, and that internal nodes of these trees not just represent some class, but that you could think of that as an ancestor that lived at some point in time of all these organisms living today. All right, so I think this is the key insight. And we now know today that all organisms are made out of cells. So there's basically with all organisms. And every cell alive today on this planet, and there are many, was the result of a cell division. Right? And every one of those ancestor cells were the result of a cell division, and every one of those was the result of a cell division. And you can take this unbroken chain of cell divisions all the way back probably to the common ancestor. Probably common ancestor of all life today. Might get a little bit mushy when we say very far back, we don't really know precisely how it works, but to a very good extent, all cells living today they're all tied up together in this sort of tree-like structure of cell divisions. So they all derive from sort of common ancestry. So that's the first point. The second point is, we know the genetic information in these cells. Okay, we know that all the genetic information. Okay, whenever I say all or something like that, in biology this is never true. All right? So there's always, epsilons here and there and things and there and there. So, but it basically says, it captures the key point. All the genetic information is actually stored in a one-dimensional molecule over a 4-letter alphabet. Okay, DNA. This is quite amazing, right? It's almost like a computer scientist made it up. So DNA is a string, and there are 4 letters, A, T, C, G. And they pair up. This rule says if you have so they're, in every organism, DNA is a double-stranded molecules. And whenever you have a C on one side, you have a G on the other. Whenever you have a T on one side, you have an A on the other. This is the Watson-Crick base pairing. And of course, it ensures that you can now nicely replicate the DNA, because if I open up this two-sided string, let's say, right? When I open up this two-sided string, I can reproduce the entire sequence by just fill it in like this, right? All right, so you can just make, you have to put a G here, an A here, a T there, right? And this is precisely the way in which organisms replicate their DNA. And we now know we have the technology to sequence these genomes. So in 1995, this was when I was in my first year of PhD, the first entire genome of an organism was sequenced, Haemophilus influenza. It has about 2 million base pairs, okay, so the length of this thing is about 2 million. And since then, there's been really an explosion in this technology. So nowadays, we have the entire genomes of thousands and thousands of organisms. And I don't think it's going to be very long until we have the complete genomes of millions of people, single-cellular organisms, anything we want. Okay? But the speed at which sequencing improves makes Moore's law look like snail's pace. Right? It's really unbelievable how much cheaper and faster these things have become. So bad that like sequencing centers and so on that used to be places where you could deposit sequence have said, please stop, we're not going to do this anymore because we cannot really handle this amount of data. I think the EBI, this is the European Bioinformatics Institute, they made some calculation. If they're going to keep storing all data sets that people want to send them, then in 15 years' time, their budget they need for that would be the entire EU signed logic or something like this, right? So the people have already years ago decided this cannot be done. There's too much data. All right. Now, of course, so you can go on the web. So you can go to, let's say, the NCBI GenBank in the US, and you can download these genomes. All right, you can get the entire genome of an organism if you want. But if you download this file, the text file, you know, what you will get is something that looks like this, right? And it just goes on for millions of pages there. There's no punctuation, right? It doesn't tell you here's something starting, here's something ending, right? So this is the problem that then bioinformaticians have to deal with. How are we going to make any sense of what's written here? So here, the fact that all these organisms come from this common ancestry helps us too, because the machinery to replicate this DNA is essentially the same machinery in all organisms, right? The details are different, but the basic molecular machinery is the same. And it turns out the way this DNA is read out is also essentially done by the same kind of machinery across all organisms. And there are two key processes. So this is called the central dogma. I might go up on that. The central dogma of molecular biology. There are two processes, transcription and translation. All right, so what happens is you have I have to get used to this thing.
If you prefer the whiteboard, that's fine.
You want to try it on the whiteboard?
Yeah.
So let's say this is the DNA. Can you see this?
Now we're going to change the page. Next page. Next page.
Next page, no, I think part of the problem is comfort of writing.
Well, I'm more comfortable writing on here. So if you can see that, that would be, for me, that would be great.
Stick to the top half.
Yes. Okay. I'll just slide it. So.
Not to talk about the center.
Yeah.
All right.
Yeah, we can leave it on actually. Eliminate this.
Press blank.
All right. So assume this is the DNA, this 1D molecule. Then there might be a chunk here on this DNA, which we call a gene. And the reason we call it a gene is because it is read out by the organism in a two-step process, where the first process, what will happen is a big molecular machine called the RNA polymerase will bind to this double-stranded DNA. So all kinds of complicated processes will happen, but what you should imagine is that a local melting of this double-stranded DNA will happen. This RNA polymerase will sit sort of around it like a big machine. And so here are letters. And what it will do is it will ride along this track. So this bubble will move along this track, and it will produce a copy, a complementary copy of this piece of DNA. All right? This is called an mRNA, messenger RNA. And so basically, whenever there's a C here, it puts a G there. Where there's an A here, it puts a T there. So it makes this mRNA string. And okay, so there is something, of course, here in the DNA that tells this machine to bind here and start this transcription. And there is something here that tells it to stop this transcription. Okay, so I won't talk about this now, but it does that. Now you have this mRNA. Now there is another big molecule, even bigger, called the ribosome, that will bind to this, and it will also move along it, along a little track, and it will read 3 letters at a time. Okay? So this may be C, T, A, or something like that. And now there will be another molecule, this is called a tRNA, that recognizes these three. It's complementary to it here. It has some sort of stuff here in between. And at the other end of this molecule is an amino acid. An amino acid is the basic molecular component of proteins. And there are 20 different amino acids. And each one of these three-letter words in RNA codes for one amino acid. Okay, so there are 64, 4 to the 3, there are 64 possible codons, these things are called. And each one of them codes for one out of 20 amino acids. So in this way, the ribosome, reading along this, translates this RNA sequence into a sequence of amino acids. All right? Now, so this is a redundant code, right? So there are many cases where one amino acid is encoded by 4 different codons. So it might be that only the first 2 letters matter, and whatever you have at the third position doesn't matter, it codes for the same amino acid. Now, there's one more, and the nice thing is that basically all organisms share the same genetic code. So this genetic code is universal. Can't not totally true, but pretty much is true. And it has 2 nice features. One is that almost all proteins, all these messages start with what's called a start codon, A to G. And there are 3 stop codons. So there are 3 combinations of these 3 letters that mean stop. And whenever the ribosome meets one of those, it will stop translating. Okay? So now this is actually the key for finding these genes. Because if you put random sequence in the genome, then with a probability 3 out of 64, you would hit the stop. And so roughly every 20 codons, you find a stop. But a typical protein has many more than 20 amino acids. And you have several hundred. All right? And so you can recognize a gene quite simply in the DNA by just looking for a stretch of codons without stops. All right? Now, of course, in reality, it's more complicated like that than that. But in bacterial genomes, pretty much that is how people find genes. They have some more sophisticated statistical models that take into account that cells like to prefer certain codons over other ones, so they don't all occur equally frequently. All amino acids don't occur equally frequently, so you can put all this kind of extra information into your model, but the basic way you find a gene is you find stretches of DNA that you can translate without stops. All right? And this has now also been extended to eukaryotes. In eukaryotes, it's more complicated because actually these genes in eukaryotes, well, I'm not even going to explain it. Okay, so don't worry about it. So we have now probabilistic algorithms that just take your sequenced string of A, C, G's, and T's and find these genes in it. And because we have this universal genetic code that translates RNA into proteins, we also know what the protein sequence is. All right? So you can go, if you go to, let's say, NCBI microbial genomes database, you can just click on one of these thousands of organisms and say, show me the genome, and it won't just show you the genome, it will also show you where are all the genes, it will give you the names of the genes, and it will even give you some rough functions of these genes. What are these genes doing? So by now, and I think this is one of the amazing accomplishments of bioinformatics, you can just go sequence something, find the genes, and even get some idea of what these genes are doing. So how did people do that? How do they find out what genes are doing? Well, let's say I take some random organism, and I have predicted where the genes are, and I translate these genes into proteins. And now I go to another organism. Let's say I go to E. coli, some bacterium that has been studied a lot of experiments done on it. I can ask, all right, so can you find me a gene in E. coli that looks like my gene? All right, so people made some algorithms if you take 2 sets of amino acids, to try and line them up to maximize the number of matches between them. So I won't go into the details of precisely how you do it, but you can write nice probabilistic algorithms where you say, well, you get some points for matching amino acids, you get some penalties for non-matching amino acids, and you also get some penalties for introducing gaps between the two, right? So you might have that you line up some letters here, and then maybe this guy has 3 more A's, and this guy has nothing here, and then they go on with the matching letters, right? So you may get something like this. So you have also some scoring scheme that gives penalties for these gaps, and so you can put this in an algorithm that in an efficient way compares 2 sequences and find the best way of aligning them together and assign some sort of score of the matching. So now I can take some gene in my organism and say, find me the gene in E. coli that looks most similar to it. Okay, that has the biggest similarity, and I will find some. I might find a gene. So if I take 2 bacteria that are sort of among the most distant bacteria in the tree of life. So, if this is the common ancestor of all life, and this is evolutionary time, and I have some bacterium organism X here, and E. coli there, I can take a gene from this organism and compare it with all the genes that exist in the E. coli genome, and find for this gene, so here's the genome of X, there's these genes in it, and now I've compared this gene with all genes in E. coli, and I found here this is the most matching gene I can find in E. coli. For certain genes, like the genes that make up these basic components, like the ribosome and the polymerase, you can go to an organism that has been separated by more than 1 billion years of evolution from E. coli, and they may still be something like 40% identical, the 2 sequences. Right? So you can clearly recognize that these 2 sequences are sort of offspring of a gene that already existed in this common ancestor a very long time. All right, so this is now a general concept, especially if you take organisms that are maybe a little less distantly related, then for any gene in your first organism, you can find a sort of sister gene in the other organism, which is called an ortholog. So what it means to be an ortholog is that in the common ancestor genome, there was some gene already there, performed some function, and as evolution happened, and these 2 organisms living today derived from this common ancestor organism, there are 2 genes in the organisms living today. So if I call this X, I might call this X1 and this X2, and X1 and X2 are basically derived through copying at every replication from this common ancestor. All right. So now, so then what people started doing is designing algorithms for finding these orthologous genes. So that if I go in this genome, I try to find an orthologous gene there. And the basic mechanism by which people do that is I take my gene of interest here, I compare it with all genes there in this organism, and I find the best match. And I do it the other way too. For every gene here, I compare it with all genes there to find the best match. And when I have a reciprocal best match, so if I find the gene X here and a gene Y here, such that X is the best match to Y of all the genes in this genome, and Y is the best match to X of all the genes in that genome, we'll assume that X and Y are an orthologous pair that derive from a common ancestor. All right?
Best match you mean maximum number of same. What exactly is best match?
Yes. Okay. So you do want me to tell you how you score alignments. Okay. So. Okay. So this is this is one of these things that people have learned over time. But right. Okay, so imagine you have your 2 sequences. And so let's call the first sequence S and the second sequence T. And so the letters are S1, S2, up to SL, and T1, T2, T3, up to TL. Okay? And now I want to make an alignment of these two. And so okay, so the first thing I need to convince you of is that an alignment of these two sequences is a path through this matrix. Okay? So let's say I want to start by aligning S1 to T3. So T1 and T2 are not going to be aligned to anything. People indicate this with gaps, like gap symbols like this. Then say I want to align S2 to S4, S3 to S5.
You align S1 to T3 because
I'm just now showing you one way I could align them. Now I could decide to go over a couple places like this, so like that. What this means is now I skip positions S4, 5, and S6. And now I align S7 to T6. All right? So all I'm trying to tell you is that a path that goes down this matrix specifies an alignment. All right? Now, typically, there is a scoring function that basically says when you align two letters, alpha comma beta, there is some probability associated with it, which is very high when the two are the same. It's still positive when they're similar amino acids, and it's not so good when they're very dissimilar amino acids. Now, every time you take a step downward, you add a gap. So there is a probability for a gap also. In fact, typically the way they've done it is there are two probabilities. There's one for starting a gap, and there is another one for extending a gap. So the probability that you assign for starting a new gap when you are matching is different than for extending one when you were already in a gap. All right? But then if I make an alignment like this, the probability of this alignment, of this example, would be P S1 T3, P S2 T4 times P S3 T5. Now I've opened the gap, so there would be a P start. Then I extended it 2 times, so there would be a P extend squared. And now there would be a P S7 T6, and so on. All right? And how have people learned these numbers? Well, it's some sort of trial and error thing where people have started with pairs of proteins that they knew were orthologs because they knew what they do, they knew what their function is, and they lined them up. And they did this many times. And then they looked, how many times do I see alpha, beta pairs in such things that I trust are orthologs? And then I built a statistical model and so on. All right, so this is how they... And okay, so the nice thing is you can find the optimal alignment, right? Because if I now give you the two sequences, I tell you how to score them. Now I want to find the best one. Or I might even want to do like something like some overall ways of aligning them. This can all be done with dynamic programming techniques. So statistical physicists might call these transfer matrix techniques, right? So the thing is, what you do is you, for example, say, to find the best alignment that aligns, let's say, only up to position I in this sequence and up to J in that sequence, right? I only need to know what is the score of the best alignment that ends here, the best alignment that ends there, and the best alignment that ends there, and then there is a score with this, extending this alignment either by matching these two or going from a gap or going here from a gap, right? So what you do is you fill in this matrix with numbers that say what is the best alignment that I can get that stops at this position and that stops in either a matching state and a gap state coming from above or a gap state coming from the left. All right? And then once you fill that matrix in, you can then backtrack what is the best alignment. You can also do this to some overall possible alignments. Right? So you could even calculate things like, I don't care about anything. All I want to know is, do letters I and J, do they need to be aligned to each other? You could sum over all paths that go through this point. And you do this again with this dynamic programming technique. So we know how to calculate things like that. All right. So imagine now that I've, using many genomes and alignment, have made an alignment of a large number of gene sequences that are all orthologs of each other. Okay? So these might be part of a protein that codes for this ribosome, and I basically found the sequence in each genome. So this is the sequence from genome 1, and this is the sequence from genome 2, and so on, genome N. And I put them all into a big multiple alignment. So this basically gives me a picture of all the sequences from all the genomes that encode this protein that has some particular function, and that are all sisters. They all came from this common ancestor, right? So if you imagine it again, there was some gene in the common ancestor, evolution has taken place, and now you've picked the copy of this gene that exists in all genes today. Now, of course, first of all, you need to make a multiple alignment, right? I just told you how you align a pair, but now I have to extend this to some large number. All right, so now that problem has not been solved to complete satisfaction, because the previous problem I showed you with dynamic programming, you can do a linear time. But if you go to n sequences, it actually becomes polynomial to the power n, because your matrix becomes n-dimensional and you'll have to look past this. So people have found different ways to try to solve this problem. And sort of one way of doing it is, first you find the best pairs. You align these pairs, you super glue those alignments together, and then you go with pairs of pairs and you put the two, right? So you first make a pair of sequences aligned, and another closely related pair of sequences aligned, and then you fix these alignments, and now you try to align these two alignments together. And then you keep adding in sequences like that. That's called progressive alignment, and it's a very popular and successful way of building multiple alignments. Another way of building multiple alignments uses consistency. And so basically what it does is it looks at all triplets of sequences. And it looks at how would A and B like to align to each other, how like A and C align to each other, and how much do B and C align to each other. And by basically checking the consistency that when a letter in A likes to align to a letter in B, A is fine. A letter in A likes to align to a certain letter in C. If the same letters like to align to each other, then you basically increase the probability that these really belong together. So this is called consistent alignment. But what I wanted to tell you about is that once you've made such a family of sequences aligned, you can represent those with a model that tries to capture the statistical properties of this entire family of genes. All right? And the way that people do that is they basically treat alignment columns as independent of each other. Okay? So what you do is, so you have here aligned to each other all these sequences, and you look at the letters that occur at this position, and you assume that these letters are drawn from some simple probability distribution, W alpha, where W alpha is simply the probability to have letter alpha at this. So let's say this is at position I of the alignment, the probability to have letter alpha at position I. Okay? This is what people call a position-specific weight matrix. All right? And if I assume that all my sequences are statistically independent, which is basically only a valid assumption if these organisms are sort of sufficiently separated from each other evolutionarily, and I assume that all the columns in this alignment are independent of each other, then I can calculate the probability of my data set of sequences, let's call it capital S, simply as follows. So the probability of the set of sequences given a weight matrix is the product over all sequences in this set, and then for each sequence a product over all positions from 1 to L, and then simply the weight matrix entry. What is the probability for having letter SI, which is the ith letter in sequence S, at position I? Now I can change the order of these products. These are all finite products. And basically you'll see that this probability really only depends on, okay, let's go up here, only depends on the number of times each letter occurs in this column. All right? So if this column can be characterized by just the number of times that each letter occurs in this column, and the probability of the entire sequence alignment under this weight matrix model is simply product over all columns, and then for each column product over all letters, just the probability for this letter at this column to the power how many times do you see this letter at this column. All right? So now we can use Bayes' theorem to give an alignment of letters, given this multiple sequence alignment S, go to what is now the probability of such a weight matrix given my sequence alignment. For that, I'm going to need the prior probability over these weight matrices. And the prior probability that people like to use in this context is called a Dirichlet prior, which simply has the same structure as the likelihood. Is basically a product over all positions in alpha, wi alpha to some power lambda minus 1. And you need to normalize this. If you normalize this, you get, so this is alphabet size times lambda. All right, and this is the gamma function. This is now a normalized weight matrix, so if I integrate this over all possible probability distributions that satisfy the constraint that when I sum over all the letters, the probabilities have to add up to 1.
The denominator is gamma of lambda or gamma of
Sorry. Somebody's paying attention. Okay. All right. So if I now multiply the likelihood by the prior, it will have this simple form: product over all columns, product over all letters, wi alpha to the ni alpha plus lambda minus 1, and then normalize it this way. Alphabet size. All right? And as Devinda already told you, the posterior is simply proportional to this product of the likelihood and the prior, and so the posterior distribution for this weight matrix given the data is simply proportional to this product. And if I now use this to calculate what is the expected value of such a weight matrix entry, you will find these integrals, these are these beta function-like integrals. It's simply given by this equation, right? So in the limit of you have a large number of sequences, this lambda from the Dirichlet prior becomes a small perturbation, and you simply get the expected weight matrix entry is the frequency of the letter in this column, which you see. So why do you have this? This lambda is also called a pseudo count, and you can see here why, right? Because it's almost like you had another count of lambda. And so typical values may be lambda is 1/2 or lambda is 1. These are typical values that people use. And what it means is that let's assume I have not observed a letter at all. So let's assume that at certain position ni for letter A is 0. Okay? Now I don't infer that the probability of having this letter is absolutely 0. I assume that it's something like lambda over n plus lambda. Right? So if lambda is order 1, this is order 1 over n. Basically what you're saying is, given my data, I know that the probability of this letter is 1 over n or less, but I can't really say that it's 0. All right? So this is the way in which this prior regularizes the conclusions you draw from this alignment. So, to just summarize this part, I didn't get anywhere near yet the work that we did on this problem. I'm just telling you what sort of bioinformatics, how they look at sequences, how they build families. Once you represented your family by such a weight matrix, you can now also use this weight matrix to go into a new genome and detect a new member of this family by basically going and saying, what is the probability that some new sequence S is drawn from the same weight matrix, which is simply this product of the weight matrix entries for this sequence. And so this is what people do. So they have built families. They know these are all proteins that have a certain function. And then now they can go into a new organism, and they look for new matches to this family. And when you represent a family like this, you can find matches that you would never detect by pairwise sequence identity. Right? So you can get pairwise sequence identity that maybe is only as low as 15%. And so statistically, you would not be able to detect this significant match between the two sequences if you just compare them pairwise. But if you compare your sequence against this entire profile that you represent by such a weight matrix, then you do detect that this is another member of this family. All right? And so what people have discovered is that they can build very big families like this. And they've discovered that sometimes, or actually quite often, you can have sequences that are statistically very diverged, so that on a pairwise basis you wouldn't detect any similarity. But if you look at the structures of these sequences, if you look at the three-dimensional shapes of the proteins they make, they are very, very similar. Okay, so what people have discovered is that protein structure evolves much slower than sequences. And that sequences that are even very far diverged often still have very similar structures. So the final thing, I guess, I'd like to say is that what people nowadays know is that actually the space of structures is fairly limited. All right? So it turns out there is only maybe a few hundred basic three-dimensional folds, and that all proteins are made out of combinations of these folds with slight modifications, that these sequences evolved but largely maintain their folds. And so there are now databases that basically have such models that are a little bit more sophisticated than I say. So apart from these weight matrices, they also have scores for adding gaps and extra sequences in, and these are represented as hidden Markov models. So they're a little bit more sophisticated, but it's basically the same idea. And so you have these large collections of such hidden Markov models, and they represent protein domains, and there might be 10,000 or something. There's a very famous collection, it's called PFAM. You can go on the web. Yes. And if you go to PFAM, you will find 13,000 of such hidden Markov models. And what people do is standard, when they have a new genome, they find the gene, they run all these models over it, they find new members from all these families, and in this way they annotate new members of this family, and they annotate their structures and sequences. Okay, so with that I'm done with my introduction. And so next time I'd like to tell you what we have worked on is to try and go beyond this model and say, can we now include the fact that certain pairs of these positions may not be independent, but there are dependencies between the amino acids that occur between these positions. How would we represent this now? How would we learn these dependencies? And can we use this to actually go from such a multiple alignment and try to infer which letters in the protein are touching each other? So can we learn something about the 3D structure of the protein from looking at the statistics of the co-occurrence of letters? So I'll tell you about this in the next lecture. All right.
running 15 minutes late. Very briefly, a couple of questions.
Hi, so you said like there is a lot of redundancy in how amino acids are coded.
Yes.
So I mean why is that? Does it have to do with error correction?
Okay, so I think the opposite of this question is more interesting. So the opposite of this question is, how on earth did this evolve at all? How did cells, or this pre-version of cells, how did they learn, how did they come about through evolution of this machinery that translates nucleotide sequences into amino acid sequences? So many people believe that there was, before there were protein sequences, before there was translation, there were RNAs, and that RNAs, they also fold up into a 3D shape, and some of them have enzymatic activity. And some people think that there was an RNA world where you only had RNAs, and the RNAs were doing everything. And at some point, they started learning making proteins. So the question is, how do you even get to a definite code? And so most sort of people that build toy models of how you might get there, they often start from the translation is random. And then over time, some, biases start evolving, and things may get more and more determined. And at some point, it froze into the genetic code we have now. And if you have 2 letters, you only have 16. You want to encode 20, so then you need at least 3. And somehow this process froze in this state where you have this amount of redundancy. I don't think it's optimal for anything. Although it is true that people have shown is that if you make errors, you tend to replace one amino acid by another one that is chemically kind of similar. So the code is not random. It has some sort of biases that sort of have some error correction properties.
Okay, any other questions at this point? Okay, if not, let's thank Eric. And let's thank you back at 11 for